So Marchantia sporophyte. So in Marchantia in life cycle there are two phases are there. One is sporophytic phase, another one is the gametophytic phase. The plant is the gametophytic one. So in this plant the reproductive organs are antheridia and archegonia are produced in different plants. After fertilization zygote develops in the archegonium. So from this zygote the sporophytic phase starts. So how the sporophyte of Marchantia is seen? So the sporophyte of Marchantia is completely depend on the gametophyte. The fertilized egg enlarges in size and fills up the cavity of the venter. It invests itself immediately with a thin cellulose wall and becomes the oospore zygote, which is the mother cell of the sporophytic generation. The zygote cell inside the venter divides first transverse wall forming and outer epibasal cell and inner hypobasal cell. This is followed by a second division at right angle to the first division forming quadrant of cells. The two upper epibasal cells of a quadrant divide and redivide and form a capsule and the upper part of the seta. So this is the sporophytic phase of the Marchantia. So here the Marchantia, this is the carpus, calyptra, perigynum. These are develops from the archegonium. So this the inside part, this is developed by the zygote or sporophytic mother cell. The sporophytic mother cell divides the first division, transverse division, and form two cells. These are divides and form the multicellular region. The two hypobasal cells after several divisions form the lower part of the seta and the foot. The cells within the capsule are early differentiated into the peripheral single layered amphitheecium and the central many celled endothecium. So you see the main parts of the sporophyte is the foot, seta and the capsule. These are the three main parts we are seen in the sporophytic stage. Or capsule of Marchantia or sporophyte of Marchantia. So this is the foot and seta are developed from the lower basal cells and the capsule is developed from the upper basal cell. So this upper basal form two types of tissues, amphitheecium and the endothecium. So all the cells of the single layered a single layered outer jacket is formed from the anticlinal division of amphitheecial cells. All the cells of endothecium and other hand functions as archisporium and forms a massive sporogenous tissue. Subsequently, about half of the sporogenous tissue by repeated divisions form a large number of spore mother cell. Each spore mother cell following the meiotic division and give rise to a spore tetrad. So the remaining cells of the sporogenous tissue function the later mother cells. So inside this wall layer is formed from the amphitheecium and the inside the tissue the sporogenous the endothecium tissue. This endothecium most of the cells are developed into the spore mother cells. These spore mother cells divide meiotically and forms the haploid spores. The remaining are act as the elater mother cells. So the elaters are formed. So the remaining spore tissue functions elater mother cells and subsequently become elongated and endowed with two spiral thickenings and become the sterile elaters. Unlike spores, the elaters are diploid cells and are hygroscopic, help in the spore dispersal. The early development of zygote is associated with several significant changes in the gametophytic tissue surrounding the embryo. The stimulus of fertilization leads to the peripheral divisions of the ventral wall and which eventually give rise to three-layered calyptra enveloping the growing sporangium. A ring of basal cells of archisporium forms second one cell thick protective covering round the growing sporangium. It is called the perigynum or pseudoperianth. Therefore, the sporangium of Marchantia develops the three gametophytic coverings Y calyptra, perigynum and perichecium. So these are the surrounding tissue, the perigynum, perichecium and calyptra. These are developed from the venter, the peripheral cells of the venter. So here the elaters, you see these are the Elater mother cells divides mitotically and form spiral thickening elaters. These are hygroscopic in nature. The main function of elaters is the spore dispersal. Now you see the mature sporophyte. The structure of mature sporophyte consists of foot, seta and capsule. A foot is an expanded bulbous mass of cells at the base of sporangium. Serves as absorbing archegonia, anchoring organ. It absorbs nutrients from the gametophytes. So here the foot is the bulbous structure. Bulbous structure, it anchors to the gametophyte and absorbs the nutrients from the gametophyte. The second part is the seta. It is slightly elongated stalk and consists of foot and capsule. The cells are parenchymatous and elongated lengthwise. The seta increases the length and pushes mature capsule out from the calyptra. So this is the seta. It connects the foot and capsule of the sporophyte. So it pushes the sporophyte, the capsule from the calyptra. The third main part is the capsule. The jacket of hanging mature sporangium, sporogonium gets dried and split longitudinally into variable number of longitudinal lobes. These lobes are extended from apex of the middle of the capsule. The capsule is a yellow colored spherical structure, contains numerous spores and elaters. The capsule is protected by the three coverings via calyptra, perigon and perigon. The dehiscence of the capsule. The jacket of hanging mature capsule get dried and splits longitudinally into variable number of longitudinal lobes. These lobes extended from the apex of the middle of the capsule. The discharged dispersal of spores to the air and are assisted by the coiling and uncoiling of the elaters due to their hygroscopic nature. The spores are almost round and very minute in size, with a thin inner intine and slightly thicker outer exine. In some species, an additional layer of perispore is also found outside the exine. So the spores are very minute. The capsule dehiscence longitudinally, and the spores come out from the capsule. So the spores are germinate and starts the gametophytic phase. So the new gametophyte. The spore is the first cell of the gametophyte which germinates to form a new gametophytic plants. Spores remain viable for about a year. Under favorable conditions, they absorb moisture from the substratum and increases in size. At this stage, chloroplast disappears transversely to equal cells. So these tetrad spores are very thin. They germinate and forms a filament-like structure. From this, the spores are two are male and two are female, develops into the two male plants and the two female plants. So in this way again the gametophyte is develops. So now you see the life cycle of the Marchantia. The life cycle of Marchantia shows the alternation of generations, sporophytic phase and the gametophytic phase. So alternation of generations are seen. So here this is from the zygote to the spore dispersal. All these are the sporophytic phases. The plant and the development of sex organs, all these are come under the gametophytic phase. So here the life cycle is the alternation of generations. In this way, in Marchantia, the alternation of generations takes place. So in this way the life cycle of Marchantia is completed. Thank you.